Hello, I'm Natasha Pinol at AAAS, the nonprofit international science society that publishes the science family of journals. I'm speaking today with Dr. Erez Lieberman Aiden, a fellow at the Harvard Society of Fellows and visiting faculty at Google. He is the new Grand Prize winner and North America regional winner of the GE and Science Prize for Young Life Scientists, an annual competition which includes an award and publication in the journal Science. Erez, in your essay entitled Zoom, you convey how your work contributes to the field of molecular biology. Could you please describe your novel approach?
If you take a pair of headphones and stuff it in your pocket, and then later you take it out because you want to listen to some music, you usually find that it's become a tangled mess, and now you've got to unentangle it before you use it. This suggests a basic mystery about the human genome, which is that the human genome, if you stretch it out, is about 2 meters long, about twice the length of a pair of headphones. And yet it has to fold up to fit inside a 5-micron wide pocket into the nucleus of a cell. How does it do that while remaining accessible? The work I and collaborators at the Broad and UMass Medical School did was aimed at developing a technique called Hi-C that enables one to reconstruct how the human genome is folded in 3 dimensions, enabling us to probe that question of how it all fits while remaining accessible.
What inspired you to devote your time to this field? What are your plans for the future?
Well, when I was an undergrad at Princeton, I remember a very striking talk by Bob Austin where he was telling some of the physics students that biology was very, very difficult because you could never really see what was going on. You were constantly trying to indirectly reason about what was happening inside of a cell. Then later, when I was in graduate school working with Eric Lander, he really highlighted for me the power that DNA sequencing has to probe a wide variety of cell processes. And so that got me thinking about this problem about headphones and about the genome. Would it potentially be possible to use DNA sequencing as essentially an imaging technology to try to give you a sense of structurally how the human genome is folded inside the nucleus of a cell?
Erez, how will this new method that you created help scientific research?
Well, it turns out that the types of structural maps that one can create with Hi-C can reveal principles that describe how the human genome is folded, even at the very coarse resolution of the initial maps that we published. For instance, one feature that we identified is that there appears to be a spatial segregation between active and inactive portions of the human genome. It's something like epigenetics via origami. The genome changes its fold in order to change its function. That mechanism is potentially very, very interesting and can help us develop a better understanding of both how cells differentiate and how that differentiation process can go awry, for instance, in cancer. Another interesting feature that we found is that our data at the megabase scale appeared to be consistent with a fractal globule, which is a previously unobserved polymer structure that couples an extraordinary density with an almost total absence of knots. So it helps explain this paradox about the headphones, how you can pack so much genome in such a small space while keeping things accessible.
Thank you very much for your interview today, Erez.
Thanks so much. It was my pleasure.